Hi guys, welcome to my YouTube channel. Today we are going to discuss a simple experiment on how to view plant cells using a microscope. Onion cells are often used as their cells are relatively big and therefore they can be seen clearly. However, they do not contain any chloroplast. If you wish to view chloroplast, you use a moss plant. The bulb of an onion is formed from modified leaves. While this photosynthesis takes place in leaves of onion containing chloroplast, the little glucose that is produced from this process is converted to starch and stored in the bulb. It is very important to know the equipments that are needed for that practical and understanding their roles. Equipment that are needed are a thin onion membrane. You need to cut small section of onion and then use forceps to peel away the inner surface. This inner surface looks a little like tissue paper. It is very thin. The second equipment that we are going to look at is microscopic glass slides. Microscopic slide is used to hold objects for examination under a microscope. The third equipment that we are going to look at is microscopic cover slips. It keeps your specimen and samples safe whilst you work by stopping any external interference from dust or other contaminants. The fourth equipment that we are going to look at is dissecting needle. It is used when you want to place or remove specimen on the slide. The fifth equipment we are going to look at is blotting paper. It is mainly used in microscope to remove excess liquids from slide before viewing. The sixth equipment we are going to look at is dropper pipette. It is used to transfer small quantities of liquids. We need also either iodine solution or methylene blue, which we are going to use as stain. They are going to stain the cells so that they become more visible under the microscope. We also need water, and then finally we need the microscope. It is used to observe the organelles found in the cell. Now we are going to look at the procedure that is followed in this practical. First, we need to add a drop of water at the center of microscopic slide. Having pulled off a thin membrane from onion layer, we are going to lay it at the center of the microscopic slide. That drop of water will help flatten the membrane. The third step is we need to add a drop of either iodine solution or methylene blue on the onion membrane. The fourth step is we need to gently lay a microscopic cover slip on the membrane and press it down gently using needle to remove air bubbles. The fifth step is we need to touch the blotting paper on one side of the slide to drain excess iodine or water solution. And then the sixth step is we need to place the slide on the microscopic stage under low power and observe the cells. We need to adjust focus for clarity and observe. So students can make another slide with methylene blue and without adding any stain to see the difference between stained and non-stained cells. So this is what we are going to observe. We are going to observe a large rectangular interlocking cells. We are going to see clearly visible distinct cell walls surrounding the cells. We'll see dark stained nucleus, and also we are going to see large vacuoles at the center. We are going to see the small granules inside the cell within the cytoplasm. So this diagram is showing how onion cells appear when methylene blue is used. Iodine solution, the cell will look like this in this diagram. And finally, without staining, the cells will look like what is in this diagram. So let us move straight to the discussion and conclusion for this practical. The layers of an onion contain simple sugars, some of which are stored as starch, which is also known as starch granules. Given that iodine tends to bind to starch, it stains starch granules when the two come into contact, making them visible. Although onions may not have as much starch as potato and other plants, the iodine stains allow for little starch molecules to be visible under the microscope. Although onions are plants, you will not see any chloroplast in their slides. This is an easy and fun activity that will allow the students to learn about cell structure. Students have opportunity to experiment and observe onion membrane with iodine, methylene blue, and without stain, and observe the differences. This marks the end of our tutorial today on observing plant cells on the microscope. Thank you so much, guys, for following me on this channel. Please don't forget to subscribe, like, and share my videos. I love you all. This is Eve signing out.